everyone. My name is Katherine Martinez, excuse me, and I'll be presenting research I assisted with this past summer titled Behavioral Studies of Ischemic Female Rats Treated with Extracellular Vesicles Derived from Human Mesenchymal Stem Cells. So a little introduction on the research that I've been doing. Strokes are the 5th leading cause of death and disability in the US, with ischemic strokes accounting for 87% of all strokes. In addition, women have a greater risk for stroke than men, with women experiencing more severe strokes with poorer outcomes. In order to optimize recovery in individuals, the timing in which treatments such as TPA is vital. TPA has a therapeutic window of 4.5 hours. So in order for to have optimal recovery, you have to administer it within that time frame. Many studies have shown that mesenchymal stem cells have great potential to promote tissue repair and regeneration in nearby cells through extracellular vesicles. As you can see in the diagram, extracellular vesicles are a cell component. These cell components accomplish this by transporting and recruiting necessary proteins, such as microRNA and integrins, to surrounding cells. Because of this and the ability for extracellular vesicles to cross the blood-brain barrier and target tissues, they have been studied as a potential mode for therapy transportation. Therefore, in this research, we want to track neurotissue recovery through diffusion-weighted imaging taken at the 21.1 Tesla magnet, which is the background I have here, and characterize motor functions to track the neural progression of ischemic female rats who received EV therapy versus the control group that received PBS. So methods. In this study, female rats experienced a middle cerebral artery occlusion for an hour to stimulate an ischemic stroke. Then the rats received either the EV therapy or phosphate buffered saline as the control group. And then we used the 21.1 Tesla magnet on day 0, 1, 3, and 7 following the procedure to capture T2 weighted and diffusion weighted images. The rats also went through various behavioral characterization evaluations on each of these days. One of these tests is called evaluated swing test, which assesses directional preference to indicate muscle and neurological deficit following a stroke. The direction the rat swings is recorded in each test. Bias to swing towards one side indicates a motor deficit. Another test conducted was the elevated plus maze. This is used to measure anxiety behavior in the rodents. The time spent in the open arms versus the closed arms is recorded to evaluate this. An open field test was also conducted. This test evaluates deficits in vertical and horizontal locomotion, and the time spent in specific grid markings and distance traveled are recorded over the span of 10 minutes. And finally, a cylinder rearing test is done to evaluate asymmetry in the forelimbs. Taps on the left, right, and both forelimbs on the cylinder wall are recorded. Asymmetrical use of the forelimbs indicates unilateral brain damage. So as you can see in the results here, the top chart indicates the percent change in the apparent diffusion coefficient, which is shown in these images. The top images is the T2 weighted images, and the bottom row of images is the diffusion weighted images. And the white part on the images is the stroke. So you can see the size of the stroke gets smaller over time. And on the bottom image, the darker areas where the stroke occurred, and it gets lighter over time, indicating that more diffusion is occurring in that area. Then the cylinder test showed that the control group scored higher overall in the asymmetry score compared to the EV group. So they had more asymmetrical motor functions. And the time in the center of the open field test, the EV and control group is seen experiencing different trends. EV decreases while the PBS increases over time, which means that the EV spent less time in the center. The plus maze, the PBS spent more time on average in the open arm, and the distance traveled in the open field, the EV on average traveled more compared to the PBS. So conclusions and discussion. Preliminary results of this ongoing study indicate that 3D EV treatment leads to enhanced recovery following a transient ischemic stroke. The apparent diffusion coefficient increases more gradually with EV therapy and begins to decrease by day 21, unlike PBS control. Smaller asymmetrical score was shown with the EV, unlike PBS control. EV and control groups experienced opposing trends for time in the center of the open field. The control group spent more time in the open arms of the plus maze, and EV group covered more distance in the open field. Further research will be required to increase sample size and obtain more accurate average results. So in this upcoming year, I will continue to work with Dr. Grant and Jamini Bagu, increasing the sample size and continuing our work and finding better ways to optimize data processing of tests such as the open field. I would like to thank Dr. Grant and Jamini and the MagLab REU for helping me with this research and for giving me this learning opportunity over the summer. Thank you.